Okay, now we are recording again. And this time I got my volume turned on. I'm going to crank. Okay, it already is cranked up. I don't know how my volume got turned off. That's the weirdest thing. While waiting for this morning's video to upload, my God, it took longer for this video to download and then upload than it did for me to make the recording. So maybe I was gaming and that caused something to glitch, who knows? And the game was nothing but glitchiness, and it was just like one of those nothing Roblox games here. So who knows? Let's go ahead. Well, I'm officially recording here. So we're going to cover cell division now. And let's get the PowerPoint up. So we got through eukaryotic cell structure this morning, and now we're going to deal with how somatic cells basically make copies of themselves. So we have this statement that all cells arise from pre-existing cells, and for the most part, they do this by a process we call cell division. At this point, I got to make a cautionary statement. Many instructors at all levels, from secondary K through 12 to the universities, will refer to this process as mitosis. A cell biologist is going to have a cow, probably have a whole herd of cows over that notion. Mitosis is actually just a part of this thing we call cell division. So my personal preference, and I always used to use the term mitosis too, like everyone else did, just because everyone else did. But at the end of the day, when you get down to the nuts and bolts of things and brass tacks, it ain't that accurate. So I've switched over to the term cell division in the last 4 years or so. So instead of saying mitosis, we are going to use the term cell division. Okay, so cell division is asexual in that it does not require another individual in order for a cell to replicate. You know, a cell cuts you off on a freeway, you cell, go replicate yourself. Be careful, a eukaryotic cell can go make a copy of itself. Us humans, cats, dogs, birds, iguanas, parakeets, goldfish, these are all sexually reproducing organisms, so you need a biological male and a biological female if you want to make babies. But cells, very specifically, somatic cells are not like that. Okay, so the way this works, the cell is just going to get bigger and bigger and bigger, and at some point, it's ready. So it's going to split into 2 new copies of itself. Now, that looks to me like a lot of dang work. Why would a cell do that? Well, 3 very good reasons why cells got to do all this. Number 1, growth. Little itty bitty baby isn't going to turn into a toddler. Toddler ain't going to turn into a little kid. Little kid ain't going to turn into a big kid unless there's quite a few years of cell division going on. Number 2, in the process of going from baby to adult, and then old fart like me, they're going to discover fabulous, wonderful things in life, like turbo Hayabusas, Gixxers, and of course, before that, skateboards, scooters, bikes, BMX bikes, skydiving, rock climbing, shark diving, big knives, all that awesome stuff that we love as little kids when we were growing up. And if you discover those wonderful, magnificent things like sport bikes and skateboards, you are going to wreck yourself a few times. So that means we need cell division for repair of damage and injury. I mean, me, I'm old. My reflexes are slow. I'll be happy with the rest of my life with a Yamaha 400 Enduro. At least it's street legal bike that can go off road. So there's that. I'll leave it to young whippersnappers to go for the leader bikes. Number 3, replication. Some cells in the body are like toilet paper or napkins or coffee filters. They just get literally physically used up when you use them. So things like red blood cells are like this. They carry oxygen for about 3 months, 4 months, then boom, they're just flat out worn out like an old beat up washing machine or an old clapped out Mercedes. And you just send the thing to the salvage yard and part it out. A lot of our cells are like that. So replication to replace consumables, if you want to think of it that way. That's the 3rd major reason for cell division. Okay. Now, this thing we call cell division can be itself divided into 2 components. First one is basically mitosis. Now, this is the term I know you all have heard, where we're going to take the cell nucleus here. You can see I'm highlighting it with the cursor, and we're going to divvy up the contents of that cell nucleus. So we got 2 copies of all the DNA, enough copies of all the other things in the cell nucleus. So we're partitioning it. And then the second part of cell division is called cytokinesis. All that jazz in the cytoplasm from this morning's eukaryotic cell structure lecture. Endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, all that stuff has to be divvied up into 2 equal parts. So that's cytokinesis. So those are the two components of just cell division. Now, cell division itself is part of a larger cell cycle where you can say cells don't die, they multiply. It's like that comedian that used to have that line, we the baby kids, we don't die, we multiply. All I remember about the guy is he like died really young in his 40s. Other than that, he had the same old routine. This is like totally a 1980s, 1990s thing, if I recall correctly. Anyway, it wasn't Sam Kinison. That was some other dude, and I totally can't think of his name. At any rate, a cell cycle, it's the time from the initial formation of a cell from cell division to the point where that cell has gotten big enough and it now divides itself to form 2 new cells. None of them died, just a little cell came into the world, got big, split into 2 new cells. Now we got 2 new cells that just came into the world. They'll get big, split into 4, 4 become 8, 8 become 16, 16 become 32. And that's kind of how eukaryotic cells can be. There's a limit though. We'll cover that soon enough. Okay, so each cell cycle, we can divide, and by the way, let's go back. Each cell cycle could take only a few days, could take a few weeks, it could take years. Some cases it might take decades. So for the most part, this is going to be on an accelerated time frame of like a few weeks to a few months for your typical cells in the human body that have to undergo division on a reasonably quick basis. But there are some that just lie in wait, they sit there, and they might not replicate every few years as the need arises. Okay, so for each cell cycle is going to be broken into 4 parts. First part, I've always called it G1 phase. Some folks call it gap phase 1, gap 1 phase, whatever. This is a period of cell growth. So there's a lot of protein synthesis, membrane synthesis. The cell is physically getting bigger. Now, S phase, all that growth and stuff stops, and all the effort is turned towards copying the genetic program, making 2 copies of the DNA. So we have the original copy, and then we make a copy off of that. Now, if the cell's going to divide, each new cell that forms can have its own genetic program. So that's S phase. Now, gap phase 2, or G2 phase, as I just called it, that's a period of time where the cell is getting ready to undergo its own cell division. Now, M phase here typically takes up 1%, 10%, 0.1% of the overall cell cycle, and M phase gets all the publicity. We will say M stands for mitosis, and of course, cytokinesis. Now, there's a lot of cells that stop dividing well, I shouldn't say they'll stop dividing in the 3rd trimester of our mother's pregnancy while we're still a baby in the womb. We're getting to be a pretty big baby, probably making our mom pretty uncomfortable at that time with all the rolling and kicking and just how big we are. But we're not born yet. And a number of our cells, a heck of a lot actually, stop dividing before we're ever born. Voluntary or skeletal muscle cells stop dividing before we're ever born. And nerve cells, very specifically neurons, not glial cells, but neurons, they, for the most part, also stop undergoing cell division before we're ever born. So when these cells stop undergoing cell division, we say they lose the ability to divide. They have undergone a process called cell differentiation, or we say they've differentiated. So cells are going to differentiate a lot over the course of their multiple cell cycles. Whenever a cell turns into something, becomes something different from the cell that created it, formed it, we say it differentiates. So we have these things called stem cells, and very early in our development, like literally just hours after our mom's ovum became an egg, and then that egg became a zygote and is becoming an embryo, we have what are called totipotent stem cells. They can become anything. So you can think of it this way. A job of a stem cell is to make more stem cells. And a stem cell that can become anything in the human body, cell-wise, we say is totipotent. Now, totipotent cells are going to differentiate over the course of several divisions, and they themselves will then become pluripotent. And now a pluripotent cell still has a lot of cell division events left for it, but it can't become anything. It can just become a limited subset of things. So I always thought of a totipotent stem cell as like an 18-year-old high school grad with a 5.0 GPA who has a free ride to any college in the nation, Stanford, Harvard, Caltech, MIT, whatever. Now, a pluripotent cell might be that same student 4 years later with $120,000, $200,000 in student loans that got to be paid back. And maybe they spent too much time partying, slamming a Jack Daniels, and doing what I did, is spending too much time in a video arcade, gaming and shooting pool. And now the GPA ain't so hot, and grad school might be out of the question. The clock of their life is going tick tock, tick tock. And to paraphrase that Metallica track from Saint Anger, their lifestyle determines their death style, tick tock, tick tock. Their options ain't looking so bright anymore. They have fewer outs available to them in life. That's how a pluripotent cell is. A totipotent embryonic stem cell can go anywhere, be anything. A pluripotent stem cell, options are limited. I was in the same boat. I remember when I was going through, I did all my general ed first, all the basic English, math, writing, composition, PE stuff. And then I had to make the decision, do I go into law school, business, laboratory science, or a field research? What do I do? Architecture, engineering? I was just hedging my bets, wait until the very last minute before I made the decision and committed. So if I was a cell, I would have stayed as pluripotent as long as possible. I probably socialized a bit too much and spent way too much time gaming, but otherwise did things mostly okay. Now, a hemopoietic blood cell, I should say hemopoietic cell. These are cells that form blood. Let me tranquilize somebody's microphone here. There we go. Mute these bad boys. I'm going to end up muting everybody while I do these. A hemopoietic cell could form any number of blood cells, red blood cells, white blood cells, thrombocytes, which make platelets. So you could call those guys or gals or just cells pluripotent. Still a lot of options available. As an example, you could get through school with a bachelor's degree of, say, straight biological sciences. You could go to work for the government, any number of research labs. Heck, you might even work for a private consulting company, a big construction firm like Somas, or what's that other big one? Pacific Architects and Engineers, Atlas Engineers. I forget what the name of the big company was. But it's a big one. Oh, AECOM, the big one that would hire a lot of people with, say, life science degrees. So you still have a wide range of options, but you're probably not going to go to law school and become a lawyer or a judge. The avenue narrows there. Okay, so a hemopoietic cell, like I mentioned, it can be any type of blood cells, and these are all the options it could become. And natural killer cells, lymphoid progenitor cells, T lymphocytes, T cells, B cells, neutrophils, basophils, eosinophils, monocytes, platelet, thrombocytes, or red blood cells. But it's not going to become a nerve cell. It's not going to become a muscle cell. It's not going to become a skin cell. Okay, so that's an example of a differentiated cell. Now, here's the problem with us being on this earth the way we are in the human condition. For some reason, our genetic code, it's like computer software. Every time you go on the internet, you're probably getting a little bit of malware and a little bit of stuff that's going to corrupt your computer code to the point where if you have a 10-year-old laptop, it probably crashes so much you just send it to the scrapyard or have someone else turn it into a Linux box or something like that for offline stuff. Our cells are good for, depending on who you talk to, 44 to 46 cell division events or cell cycles. With each repeated replication of our DNA in S phase and then the subsequent cell division event, our genetic code gets corrupted. It gets damaged. Errors are made when we're copying the stuff, making copies of it. Now, we have these sequences of genetic material called telomeres at the end of each chromosome, at the end of each DNA molecule. These telomeres protect our DNA from damage when it's being copied. But over time, those wear down. Telomeres are like tires or brake pads in your car. You use them up, you wear them out. Maybe you have some hard Continental tires or some soft Michelin Pilot Sports or Hankooks, whatever. It doesn't matter. You're going to wear those tires out over time. And even if the car is a garage queen parked in the garage, they'll still rot. That's how our telomeres are. Over time, they get shorter and shorter. They wear down like tires or brake pads. And then one day, there's nothing there. It's wore down to the treads, wore them down to the metal. So when that happens, then our aging process just accelerates. Boom! And we go to hell in a handbasket. The DNA gets chewed up at the ends. And then we get non-viable cells, non-functioning cells, stuff sags, falls apart, we heal slowly, we lose our oxygen carrying ability, and then we die. And that's just a bummer. But I've been told it beats the alternative of dying young, so there's what it is. There is stuff on the internet. People are saying exercise will have the effect of lengthening telomeres. I would like to see I know there's a peer-reviewed research where they studied that and found it to be the case in rats and mice. Intermittent fasting, going keto also seems to help for a lot of individuals, but it's not going to stop the process. It's merely going to slow it down. It's going to apply the brakes to the process of aging it back. So there's that. Okay, now cell division in the cell cycle, overall, they're regulated by proteins, a lot of proteins, different proteins. Where do proteins come from? Ribosomes make them, but the genetic code, genes code for those proteins. They're the instructions for building a specific protein. So two of the proteins involved with cell division, well, not just cell division, the cell cycle as a whole, are what I call CDK, cyclin-dependent kinase, and something called cyclin. So kinases, first of all, are a class, a family of proteins that like to boss around and tell other proteins what to do. In this case, this is a kinase that will not do its job unless it has cyclin dependent. If I was an enzyme, they would call me caffeine dependent Mike, because I'm not going to get diddly squat done until I have my coffee in the morning. Then I'll think about being woken up and not being grumpy. I add a splash of Baileys to my coffee when I wake up happier, faster. Oh, and add some heavy cream too. Got to fortify this stuff, make it absolutely thermonuclear colliding black hole antimatter explosion strength coffee. Yeah, and I can get stuff done, like do video recordings for Zoom. Okay, and waste all that other rest of my energy gaming. Okay, so these two proteins, CDK and cyclin, they're going to join to form what we call MPF, mitosis promotion factor. And MPF, we got to have enough of it at certain times in the cell cycle. Very specifically, we got to have enough MPF at the end of gap phase 1. If we don't have enough there, the cell is not going to undergo division. It will undergo a permanent non-dividing state called G0 phase. Okay, so we get an idea as to what regulates this, how it goes. Now, mitosis or M phase, here on this little diagram, I just downloaded this, I just pulled this one off the internet and then edited the colors so it didn't clash so bad. It was more interpretable on a small smartphone screen or a small 5x7 tablet screen. Let me just double check participants here. Everybody tranquilized on the microphone department? Sweet. Obviously, interphase and mitosis, as you see from this diagram here, are equal. In reality, interphase is 10 to 100 times longer, bigger than mitosis. So this graph you see here, again, it's not 100% anatomically correct. I did it in some textbook, and there's content was on the internet, and I just right-clicked and cut and pasted into this PowerPoint. Okay, so as interphase really G1 phase progresses, the concentration of cyclin rises, activates all the cyclin-dependent kinase in a fully operational potent MPF, and boom, we have cell division going on, mitosis and cytokinesis, and this MPF, well, specifically the cyclin, is used up towards the end of mitosis, and it's all gone after that. During G1 phase of the next cell cycle, we got to make more of it. Okay, so that's that. Now, mitosis, cytokinesis, M phase itself takes up easily less than 10%, maybe less than 1%, depending on which textbook you're using, where you're taking this class, who your instructor is. Yeah, there's 4 to 5 phases of mitosis, and we call these the 4 or 5 phases of mitosis. And in chronological order, I just use the 4-phase system unless I'm teaching this class for Cal States or UC, then I'll go to the 5-phase scheme. So prophase, metaphase, anaphase, or telophase, PMAT, PMAT. If you're taking this course at Cal State Dominguez, then your instructor will probably talk about prometaphase. It's somewhere in between prophase and metaphase. And again, this isn't Cal State Dominguez, so we're not, or any of the Cal States really, because they all pretty much seem to use the same textbook. So we won't go for that here. Interphase is just a term for the combined G1, S, and G2. What's wrong with that there? Let's fix that. G2 phase, a combined G2 phases. Okay, since I just had to change that, let me just save this file real quick. A little typographical error in there from when I was cranking these out right after the lockdowns. Okay, so interphase by far here is the biggest component of the cell cycle, and then my prophase of mitosis takes up a fair amount of the M phase, and then metaphase, anaphase, telophase, the MAT part. Combined, they're less than 1% of the entire time for M phase by itself. So they're going to be less than 1,000th of the overall cell cycle. So if you look at this, if you look at a cell division slide, and if we were meeting in person, the rest of this lab, you'd be looking at plastic models of cell division and real fish egg slides where there's a bunch of cells undergoing all these phases. You would see that the overwhelming majority of the cells are not actually doing anything. They're somewhere in interphase, G1, S, G2, and then only a small minority, maybe 10, 20% of the cells would actually be in a part of cell division here, mitosis. Okay, and of those 10% of the cells in mitosis, probably 70% of those would be in prophase, 2 or 3, 20 or 30% might be in the metaphase, and then maybe 10% each in anaphase or telophase. Okay, so if we were to look at this stuff under the microscope, most everything will look like this, just the way you see it here. You have the cell membrane, you have cytoplasm, you have a visible nucleus with its own nuclear envelope and nuclear membrane, which is actually a lot going on there. And these cells, if they're professionally prepared and looking good, you'll be able to spot the nucleolus easy peasy lemon squeezy in these. All right, so maybe a tenth of the cells will look like this. First of all, what's going on with the nucleus here? It looks like it's disappearing. And then I see squiggly purple stuff forming inside forming chromatin or chromosomes. And then I see these little fibrous pink looking starbursty structures. They have a satiny texture. These are called centrosomes, and there are 2 of them. So in prophase, or specifically mitotic prophase, we're going to see basically these 2 centrosomes splitting apart and going to opposite ends of the cell. One goes north to see Santa Claus, the other one goes south to Antarctica to see penguins, if you want to think of the cell as like the planet Earth here. And then this purple squiggly stuff in here, that's our genetic material, but very specifically, it's a generic cocktail, a mixture of DNA and proteins we call chromatin. Now, as chemicals go, chromatin isn't like alpha glucose or benzene or 1,3-dihydroxyacetone phosphate. The chromatin is just a generic name for a mix of DNA and proteins. So there's going to be a lot of variability with chromatin here. At any rate, the stuff is going to coil up, condense into compact visible form we can see with an entry-level student-grade microscope. Of course, about that, today's entry-level student-grade microscopes are like today's entry-level beginning price point cars. A basic Honda, Toyota, Nissan, Ford in the year 2019, 2020 will outclass a 1970s, 1980s Ferrari almost in all respects. So today's student grade microscopes are way better than the stuff, even the nice things, the high end stuff I was using as a grad student. The full featured, update, modern optic manufacturing techniques, everything about them is better. Okay, so we might say you guys are looking at these with entry level microscopes, but what you would have and work with in our new building if we were meeting in person is actually really, really, really good. Cal State Dominguez, they also have new updated microscopes, but ours are even newer and in many ways better. Okay, so what we call chromatin condensation occurs in prophase. So this is sort of how it works here. Basic DNA can be stretched out. Now, that's just like a big, long string. This string is going to be coiled around these proteins called histones. There's actually 8 histones, 5 different types. We got 1A, 1B, 2A, 2B, and then a fifth one here. And they form this beads on the string structure, where you got the string being the DNA, and this little cluster of 8 balls of proteins plus one in between the 2 balls of 8, which is the fifth. These little beads are called nucleosomes. Okay, and then we got this little coiled DNA around the nucleosomes. We further wrap that into this little coil spring shape. That's called a 30 nanometer fiber. Once DNA is in the 30 nanometer fiber configuration, it's like the code is locked up. It's inaccessible. We can no longer read it to follow it and make proteins. So we're temporarily locking up the genetic code here during cell division. So it's going to be temporarily inaccessible. Kind of like when Canvas or some other internet service goes down for maintenance at midnight on a Saturday or a Friday and comes back online at 4:30 in the morning the next day. Well, this site is temporarily under site maintenance. You can't access it. Same with our DNA when it's undergoing cell division. Okay, so then this 30 nanometer fiber is going to be wrapped around into an even bigger coil, and this is called supercoiled DNA. And then the supercoiled stuff is looped back and forth into what we call domain loops, and then those are packed into this linear oval shape here. This thing we call this physical depiction of a chromosome down here, I'm circling it with the cursor, that's actually not too far from how they actually look in real life under really high magnification with a spectacularly good microscope slide. So this discrete identifiable little package down here, 2 chromatid arms attached at the middle to the same centromere. This structure is called a chromosome. Us humans, we have 23 different kinds of chromosomes, 2 copies of each for a grand total of 46 chromosomes. Here's how I like to think about chromosomes. You're a complicated creature. So is everyone else. Us humans, we're complicated biological machines. We have a lot of moving parts. A lot of things can go wrong. If you buy a washing machine, a refrigerator, a smartphone, a car, all these things come with a user's manual as well. Most of the time, we don't read the user's manual, but if you've ever noticed, the bigger and more complicated the appliance you're buying, the bigger the user manual. I know my cell phone came with one about the size of a Gideon Bible. I'd never read the user's manual. I just started swiping immediately, and it was all intuitive. A refrigerator, not so much. So us humans, our user manual is big. It has 25,000 different instructions in it, and we have 23 chapters. Luckily, we have 2 copies of our user's manual in each cell, so we have a grand total of 46 chromosomes present in each ready-to-divide somatic cell. Okay, so that's it. Prophase started. By late prophase, what some will call pro-metaphase in some textbooks and other colleges, this chromatin is fully condensed, and on a spectacularly good microscope slide, all 23 chromosomes might be visible. There will be 2 copies of each, of course, so you'll see 46 chromosomes in there. Might have to separate them out a bit, but every now and then you get a really good slide, and they just pop into view at high power magnification. So each chromosome has what's called well, let's just deal with the kinetochore. The centrosomes, one went north to see Santa Claus, the other one went south to Antarctica to see penguins. They're at opposite poles of the cell here. And they have these little microtubules, elements of the cell cytoskeleton repurposed here, coming out for them, running from one centrosome all the way across to the other centrosome, and then some that just run to the chromatin, the chromosomes themselves. Those are also microtubules. So we call these spindle fibers. These microtubules make up these spindle fibers. I don't really have it listed here, but the ones that run to the chromosomes are called kinetochore fibers, and the ones that just run from the centrosome are called polar spindle fibers or polar microtubules. Now, these centrosomes, not every textbook has used the term centrosome. Don't get it confused with chromosome. Obviously, they're not the same thing. Centrosomes, basically, there's 2 of them, paired centrioles at right angles to each other, and you saw these on the eukaryotic cell lecture that preceded this one. Some people call them asters because they look like these little flower star-like structures. Others call them MTOCs, microtubule organizing centers. Whatever term you like, go for it. You know me, kickbacksadasical. Metaphase, and I invented that word. It's a portmanteau of kickback and lackadaisical. So if there's a term you learned elsewhere in the world, I know a lot of my students, they might have their nursing degrees from Cameroon or Nigeria, and then they have to come over here and they have to take all the classes all over again to get certified, even though they know all the stuff. And that happens. When I was a grad student, the Soviet Union collapsed, and I had a bunch of Russian rocket scientists and nuclear physicists coming over here taking my classes at the 4-year university. And it's like they already knew this stuff. They took this stuff when my dad was my age at the time, and they wanted to emigrate to the United States and get a new start in a non-communist country. Kind of what they had to do. They had to take the classes here, and it's got to feel rough. Anyway, in metaphase, the chromosomes have all lined up on this flat equatorial plane. But anyway, speaking of folks that have learned this stuff elsewhere, the terms you learned in Russia, the Soviet Union, Ghana, Nigeria, I'm fine with those terms in my class. I know they exist. They may not be in American textbooks, but I'm aware of them. So if you use them to answer the stuff on my exams, my assignments, you get full credit. Okay, so let's get back to the topic at hand here. In metaphase, the chromosomes have all lined up on an imaginary flat plate, or on a flat equatorial plane. This is called the metaphase plate. And they're lined up in such a way that each sister chromatid or just chromatid that creates 1/2 of a fully duplicated chromosome faces the opposite direction. So each chromosome straddles the metaphase plate, centromere is at the center. One chromatid faces north, the other chromatid faces south. Now, this complete assembly here of two centrosomes, all the spindle fibers, the kinetochore microtubules and the polar microtubules together, and the chromosomes at the center, this whole thing is called the spindle apparatus. Okay, now in anaphase, the chromatids separate from each other. So the sister chromatids separate, the centromere splits, and on each side of the former centromere, there's this little structure called a kinetochore. If I had to compare a kinetochore to anything in real life, it would be a fishing reel on a fishing pole or a winch on an off-road vehicle like a Jeep or a Land Cruiser. It just like pulls things up. And basically, these chromosomes with their centromeres and kinetochores attached, think of the chromosome as like a Jeep or a Toyota Hilux or a Land Cruiser, and you got the winch on the front bumper, and that winch is like the kinetochore that hauls the vehicle out of the mud onto a hard, dry, firm road. You know, that's why you have winches. You can tie your vehicle to a tree or something and pull yourself out of the mud a little bit more easily if you do serious off-roading. Okay, so that's the kinetochore there. So the kinetochores pull the chromosome they're attached to towards the destination, either to the north here or to the south down here. As you see, I'm circling on the screen with the cursor. So this divvies up, partitions the genetic program into two identical copies. So each new cell has all the genetic material it needs to properly operate as a cell. Okay, now telophase, last part here of actual cell division. The chromosomes have reached where they're going, their destination, and a new nucleus starts to reform around each new group of chromosomes. The spindle apparatus completely disappears and becomes cell cytoskeletons for the two new cells. Circular groove called the cleavage furrow appears surrounding this cell, and this cleavage furrow is created by what's called a contractile ring, a ring of actin microfilament protein that tightens up and pinches the cell in two. So you can think of this cell as a big, soft, partially filled water balloon, tie string around the middle of it and pinch the water balloon into two parts. And since the water balloon's not very full, hopefully it won't rupture and spill all over your pants while you're doing that, while you're demonstrating that. Okay, and while this is happening, the chromatin is going to uncoil and spread out into its normal form in the cell. So then we won't be able to see it anymore. Okay, so a little thing on some genes and well, this isn't really genetics. This is just straight discussion of the genes, because genetics itself is kind of like the study of heredity and how parents pass their biological traits on to children. When we talk about genes and the proteins they code for, a better term for this would be molecular biology. So a little bit of molecular bio here. Everything in the human body, for the most part, is regulated by genes that code for proteins. Cell cycle is no exception. So the genes that regulate cell division by coding for proteins like cyclin and cyclin-dependent kinase, actually, there's at least 4 different types of cyclins, and they're all important here. We call these proto-oncogenes. So a proto-oncogene just codes for a normal protein. Cyclin-dependent kinase involved with normal cell division, normal growth, repair, and replication. But like any other gene, they can mutate. The genetic source code can be corrupted when a mutation happens. Most of the time when this happens, nothing happens. It's just like a bad computer file that doesn't open anymore. So you got to reload it, reboot it, download the patch off the internet from whatever, or maybe the Microsoft or Apple will fix it with their next update, and boom, you're good to go. But sometimes when they do mutate, they become a very bad version of their former selves that doesn't behave like it should. And we can have cell division switched permanently on to where it never stops. Now, the mutated sequences of DNA that code for these misbehaving proteins that regulate cell division, we now call these bad boys oncogenes. So oncogenes code for defective proteins that result in nonstop, runaway, uncontrollable cell division. That means you're going to get a ball of permanently dividing cells forming in around the spot where the original one mutated. And we're going to call this massive cells, which you might be able to feel with your fingertips. Definitely it's going to show up on an MRI, that's for sure. We call this mass a neoplasm. That's the new term. I grew up, these were called tumors. Okay, so a tumor that stays in one place and doesn't spread around, we'll say it's benign. You know, living with a benign tumor, to me, is like living with a roommate that likes to dress up in a clown suit, listens to Insane Clown Posse and is down with a clown, holding a chainsaw in their hand, and then they like to rev it up, go zoom, zoom, zoom. And the worst part is they like to stand up over your bed at night while you sleep, watching you sleep. I have Mr. Glock for that. My view of benign tumors is I want to get those suckers surgically cut out ASAP. And if they're on the skin, frozen off, lasered off, whatever they need to do, get rid of those things stat. I'm not one to put up with benign tumors. Why? Because we can have another mutation where the tumor changes its characteristic and now it spreads everywhere. When a tumor spreads, we say it I hate this word. Uh-oh, I don't want to do that. We say it metastasizes. So little cells will break off from the big tumor, roll around in the body. They're dividing and mutated themselves. So they'll form new tumors everywhere they go. And they'll start taking an express route in the bloodstream, going everywhere. So lymph node cancer can spread to the spleen, the liver, the heart, the brain, the muscles, arm, legs, everywhere, as an example. When they do that, we call these bad boys malignant, and that's the problem, because then that cancer is spreading. Okay, so that's all I really wanted to say about cell division and cell cycles here. Before you go play Fortnite, I don't know if I'm going to play Fortnite. No, Steam now has Fallout, the latest version of Fallout on sale. And I've heard online that it's not bad, and it's like $11. I might play $11 for the latest Fallout version and give Fortnite and all the other stuff a break. We'll see. But before we go do that, let me stop the share. And I need to go to the internet window here, go to this, and let's go to home for Canvas. Now let me go back to Zoom. Hold on, waiting for my window to pop up here. Let's go share with the home page for Canvas here. Okay, so a lot of this stuff you won't see because I have it in the lecture shell, but it's actually in the lab section. So I showed the amoeboid movement earlier. Where in the cell division lecture, I had this PowerPoint. I have a little animation here. You can check it out if you want. Here we go. Cell division images. This is what we would have seen in class. I need to minimize my Zoom window here. If we were looking at the microscope slides in our classroom in real space, pre-COVID-19, these are some of the things we would see. And in this case, the cell I'm circling here with my cursor, interphase. See a cell nucleus here, but not a whole lot going on. And then look at this cell I'm circling here with my cursor. See a microtubule organizing center or centrosome, one here, one here. You see the fibrous texture here I'm indicating with the cursor? Those are the protein, the microtubules, the spindle fibers that form the spindle apparatus. And this dark purple material here is DNA. These are actual chromosomes. Not human. This is from a fish egg, a freshwater fish they get out of places like Michigan. They call it whitefish. Tastes okay. You go eat in the restaurants in Ohio, Michigan, Indiana, Chicago, Milwaukee, and Cleveland. This is the type of fish they'll serve here. Here in California, we got the Pacific Ocean. You get saltwater fish here. Anyway, we got another cell down here showing the same thing. Let's move on. Here we go. This big cell right here, we can see the outline of the cell membrane here, the outline of that cell, a nucleus here, nothing going on here. This is an interphase, but look at this. We see a line of chromosomes with chromatid arms, beautiful, perfect textbook metaphase. See the spindle fibers, the two centrosomes, the spindle apparatus there. Another beautiful metaphase cell. This one, the chromatids have separated and are starting to move in opposite directions. So this to me is kind of looking like anaphase here. This cell right here, I'm circling it. I'm starting to see stuff forming here. I'm wondering if this isn't prophase. What else do we have here? Oh, here we go. Very nice. Look at this. Look at these two. Two new clumps of chromosomes. Things are starting to dissociate. I see this line here forming in between these two nearly identical twin cells. And then these two look to be a little bit earlier in the same process. This little line is a contractile ring. And you see this surface groove here between these two forming. That's a cleavage furrow. Same thing here, but it's just a few minutes, maybe a few hours earlier in the process. So in actuality, probably 15 minutes to an hour earlier, this cell might have looked like this one. So we see the boundary of the cell membrane here. We see a cleavage furrow. We see the chromosomes have pulled apart from each other. I don't see a whole lot of spindle fibers in between them, and I see the beginnings of a new cell partition here, a new membrane separating these two cells. I don't really see a nucleus reforming yet. These chromosomes are still well condensed compared to these here, which are kind of dissipating. But both of these look like early telophase and late telophase. And then we got some surrounding cells where nothing appears to really be happening, just pretty much interphase. Okay, so that was that. Now, I canceled this lab exercise because it takes a lot of time, and us doing this class in 4 weeks is insane. What I'm going to do is I'm going to head back to the home page here, and let's go to the lab section. So 13579, here's lecture. 13579 is lecture. 13580 is lab. So this, of course, is a whole different class, the LACCD district, which is like Mission College, Pierce College, LA Trade Tech, East LA campus, Harbor College, us, West LA campus. It's all one district. We separate anatomy into lecture and lab. Usually the same instructor teaches both, but it can be team taught by separate instructors, but you get one combined grade. If we didn't do that, the single interface Canvas shell for a combined lecture and lab in this class would be insanely busy just to look at. So I have the lab modules and their instructions here. Let's deal with module 2 here. Animal cell worksheet. So this applies to what we saw this morning. This particular worksheet here, I need to make this black and white so it's printer friendly, because what you're looking at here is not printer friendly. You could see something like this on the lab exam or quiz. Let's go back. Let's click the back button. I'm giving everyone a preview here. Let's see, lab quiz number 2, cell structure. Let's preview this. So, oh my God, where did you see this? This is the same picture, the exact same picture I had in the PowerPoint. So this is the newest Stako cell model we have. These are nice. The plastic isn't chipped, paint's all nice and fresh on them. So the way this works is, organelle A, what is that? You have a drop-down menu, and these are your choices of answers. It could be any one of these, so on and so forth. Question number 2. The organelle indicated by C is, let's see, where is C? Here it is. Oh, that little thing. What might it be? Oh, and what happens at C? Just a bunch of stuff. I don't know. You tell me. It's only one of those. And then the one indicated by D is so on and so forth. So this is basically what we would spend some of the time in lab looking at, these big plastic models here. So this is sort of the eukaryotic cell structure quiz. And this is found in the lab. Let's go back to the home page here. And let's see what else we have here available on the lab shell. Cell division model worksheet. Okay, so these are the plastic models we would refer to in a room, and we'd spend time, studying these. Now everyone has the smartphone cameras you guys are all carrying in your pockets are equal to or better than what would have been a $5,000 DSLR camera when I was going through. So these images that you get with a cell phone camera are top notch. I just have everyone take pictures of the models and the answer keys, and that way you can study this stuff anywhere, anytime, pre-COVID-19, before we ever went remote here. Anyway, I just made detailed breakdowns of what's going on with the plastic models that you guys otherwise would have viewed. You could totally easily see some of these on a quiz or an exam, guaranteed. Let's see what else we have. Always good to preview stuff. Ooh, that cell structure, we got through all of that. I am going to take you ahead. Those who are on this video, I'm going to go ahead here. Let's go to this quiz. Lab exam 1. This is a preview of your first lab exam. So we got a PowerPoint here. Now I have this modified, so things are a bit different. Oh my God. Well, you can see the body regions worksheet from basically yesterday and today. Well, today is here. Actually, today is June 15th, Tuesday. So I'm actually pre-recording late Wednesday material with this Zoom session. But anyway, this is a quick preview of a possible lab exam number 1. So histology, we'll be covering that. I'll be pre-recording stuff for histology, maybe tonight, maybe tomorrow, and officially we wouldn't start this till Thursday and we'd finish it up on Monday. Abdominal quadrat. I really, can I expand this? I want to expand this out this direction. You know what? I'm not happy about this. I want to see an alternative format. Nah, I'm just trying to make my image bigger here. So what regions? Oh, what surface of the hand? Is it palmar, plantar, dorsal, ventral? What words describe what surface of the hand? And then the feet are also very complex. So is that the palmar surface? Is that the palmar surface? Is that the plantar surface? Is that the plantar surface? What surface A and B? And then we have the various structures and phases, more of the model worksheet, and stuff from histology and the integumentary system or integument. Okay, so your current lab exam, I grade them manually by hand. The reason is the computer software for Canvas doesn't recognize all the possible answers out there, and it would be practically impossible to program all possible good answers. It won't even recognize the same word if it's capitalized versus uncapitalized. So we got that. Quick preview of exam number 1. Okay, so next up is histology, and then the integument system, and then that's our exam. So I'm going to stop share, wrap up this recording, and I'll see you next time when we start